I think we're on time. Our next speaker is Dr. Habiba Al Safar. Dr. Habiba is the director of the Biotechnology Center and an associate professor of genetics and molecular biology at the College of Medicine and Health Sciences at Khalifa University. She received her PhD in forensics and medical sciences from Western Australia and a master's in medical engineering from the UK and her bachelor from the US. She is a member of CAG's executive board. Her title of the talk is A Population-Specific Major Allele Reference Genome from the United Arab Emirates Population. You're welcome, Dr. Habiba.
Bismillah ar-Rahman ar-Rahim. Thank you, Dr. Riyad, for the great introductions. Hi, everybody. Good morning. I would like to thank the organizer for giving me this opportunity to talk about the Center for Biotechnology Center at Khalifa University and our work for the UAE genome. Our aim is to improve our understanding of the genome of the people in the United Arab Emirates. Personally, it was a journey that started before I decided my PhD studies. The journey was motivated by a personal interest and one that shaped my professional career. Diabetes was it's managed in my family, and this has been driving my professional interest. So I don't have anything to disclose. So I have 3 agenda items today. Firstly, I would like to talk about why it's important to study the genome of the United Arab Emirates. And then I will touch on the findings which have been published. And I would like to round the presentation by talking about where I see the future work is required. So the journey started when I visited Lori Hood's lab in 2017. And many of you guys know Lori Hood was one of the principal investigators for genome sequencing technology, and he is one of the inventor for multicolored sequencing machine. So since Dr. Greg Venter first published the first complete human genome sequence in 2001, addition of science sequence technology has been vastly improved. The first human genome took 10 years to complete, and the whole genome now is performed in a matter of days. Technology now makes larger scale genome studies as possible. Thirdly, through large population-based sequence project, research has uncovered variants that can be used to predict likelihood of diseases. We know that particular variants are responsible for cancer, sickle cells, cystic fibrosis, and other. Simple tests can be developed using genome data to screen and diagnose the diseases. So despite the efforts of the sequence human over the last 30 years, some populations have been poorly characterized. And in 2019, Nature paper described the results of the distribution of the genome data in the public domain. Despite progress being made in other non-European countries and population from 2009 to 2016, genome data remained predominantly from people of European origin. Importantly, from the perspective of the people of the Middle East, there was very little data. In 2016, less than 1% of the genome data in the public domain were from the from the people of the Middle East. So it became clear to us that we had to improve our numbers in the public domain. In 2018, we proposed an initiative to study 1,000 Emirati genome. So I would like to spend the next part of the presentation on what we have achieved. A year after the proposal, we published the first whole genome of Emirati, of UAE national. In fact, we sequenced 2 individuals shown there as It's shown here in the slide as UAE S001 and UAE S002. To make sure we had a backup, that's why we used two samples. In the Venn diagram, we show the number of genome variants, both UAE subjects compared to other of Kuwaiti, of Persian ancestry that was sequenced by research team at Desman Diabetes Institute in Kuwait. Although there are many variants commonly shared by the three individuals, there are also a variant that are unique to each individual. So a year later, we published another for a complete genome of UAE nationals, noting a similar findings. The four samples shared 1.4 million variants, but had unique variants that ranged from 800,000 to 1.2 million, confirming the finding in our first study. Both shows a previously unappreciated population diversity among Emiratis and potentially among people throughout the region. So it is important to delineate the association between genotype and disease of personal genomes. We relate the variant with a potential susceptibility to list of diseases. This figure shows the pathogenic and risk variants among the four samples according to Klimbar. The variants include susceptibility to cardiac defects, asthma, atopy, lumbar disc diseases. All have high values of UAE specific allele frequencies falling between 0.5 and 0.8, making them common variant for the UAE populations. Although the numerous diseases association were unique to each of the four genomes, most were not those which are highly prevalent in the UAE, such as diabetes, asthma, and cancer. So earlier this year, we published the version of the UAE reference genome. It was generated by integrating variants with the major allele frequencies of greater than 50% that were identified in the cohort of 153 UAE samples with an human genome 19 reference genome. This resulted in constructions of the UAE reference genome by replacing the 2 million site in the HG19 reference genome with the major allele calculated for the UAE for both SNPs and indels. The UAE reference genome was subsequently tested using 4 genome samples that were not used for constructing the reference genome of Emirati. The variant for each of the 4 tested samples were called by running the same variant calling, the pipeline that we used, using HG19 and UAE reference genome separately, of course. The variants for, so although no significant change in the read per locus coverage was observed, the alignment's quality improved with an increase up to 5.3 million base pair that were mapped to the UAE reference genome in comparison to the HG19 reference. This provided a greater statistical power to variant calling, where a reduction of 1 million called variant per genome when using the UAE reference genome in comparison to HG19. So this table is basically a numeric version of what the graph that I explained in my previous slides. So our analytical pipeline did not exclusively rely on a whole genome sequence data. We also used information generated from microarrays. Microarray data allowed us to analyze larger cohort as the next generation sequencing effort continued in our lab. The bar plot in this slide is generated by a mixture software tool. We used 758 individuals and ordered them accordingly to the arrangements obtained from the phylogenetic tree that we constructed. We can see the Emirati population, predominantly Middle Eastern in green, with the next largest influence from her historical population in Central and South Asia in purple. We can also see some contribution from Sub-Saharan African in red and Europe in yellow. There is only a limited North African contribution in blue to the Emirati gene pool. As expected, Native American, Oceanian, and Far Eastern contribution were very limited. So in this principal components plot, analysis plot, of the data shown in my previous slides, each Emirati samples is outlined. They were mapped to the PCA plot and construct to samples of a human genome diversity population, which cluster in relatively a very tight group. The UAE subjects fall entire spectrum, losing around the intersection of these two axes. This loses clustering, showing a continuation in the distribution of samples, suggesting a gradual diversity across the Emirati population. Each UAE subject is representing with a pie chart to show the ancestral composition of the individual determined by the admixture software. The pie charts largely correspond to their mapping of the principal component. So what does all this mean? The genetic composition of these people, of the people of the United Arab Emirates, has been influenced by events over centuries. Travel was relatively common as a population migrated for trade in pursuit of knowledge. Different population intersected with where the admixture occurred. Over the time, it has given rise to the current population that lived throughout the Middle East. So we are looking forward, and we started, we started the project in 2006, over 15 years ago, when we enrolled our first participant into Emirates Family Registry. When I started working at Khalifa University in 2011, we established a DNA lab. We continued to collect samples and upgrade our Sanger sequencing to next generation sequencing in 2014. To consolidate the genome science at Khalifa University, the Center for Biotechnology was established in 2015. We added a bioinformatics division about 5 years ago, and the work I have just described has resulted from this journey. Our next challenge is to really relate the key variants of the Emirati genome to a clinical outcome. So at the Biotechnology Center, our primary interest is cardiometabolic disorder. We have collected DNA from families with diabetes and heart diseases. We prefer to study families rather than individuals. Families simplify the analysis process as it provides more information on phase. We have pairs of chromosomes, and family studies allow us to differentiate between the maternal and paternal contributions. We apply ranges of technology. As mentioned, we started with microarrays. We currently use whole genome and whole exome sequencing in our work. It's also important to collect the phenotypical data. After all, we are studying disease. The deep phenotype data will make a very common sense on your results, basically. So I have shown data that shows that there is a high level of diversity in the Middle East. There are different definitions of what we are made of in the Middle Eastern region. We know that the population of Arabian Peninsula is influenced by human migration out of Africa and with Europe and South or Central Asia. Using the Middle East and Northern Africa or MENA economics region as definition, there is a range of ethnic groups that we need to study and to appreciate the diversity of this region, basically. To improve the diversity or to improve the delivery of the healthcare in the UAE, we have to appreciate the relationship with our neighbor who have already shaped their population and implemented the genome medicine. To improve our rate of discovery, collaboration is a major factor. As my colleague, Dr. Khalid mentions this, it's important to develop centers for research excellencies. As each population have different diseases profile, it is important to focus on the local problems. Technology is no longer a rate of limiting step. Once it was prestigious to build a genome lab. It is not a factor to generate data. It is important to build technology hubs that we can all send samples or for rapid sequencing. For faster, we can collect data, the quicker we can perform analysis, and the sooner we will find solutions. As a group, I believe it's important to promote staff and also student exchange with our neighbors countries. Once we have strengthened our local and regional competencies, then we can we can contribute to the international effort to improve our understanding of genomics. As I mentioned, I believe the future requires to improve our understanding on how genes contribute to disease. To do so, it's important to establish longitudinal studies. We must collect time series data so we can relate variants that develop and progress of disease. Significant and long-standing effort exists around the world, like Busselton Group and Birmingham study, which is one of the famous longitudinal study, which started in 1948. Which is the Birmingham Heart Study. And research from this longitudinal study had significant impact on human health. Its finding has included the discovery that high blood pressure, diabetes, and cigarette smoking are a key contributing to heart diseases. All developed countries have developed major longitudinal study, and there should be a coordinated effort to build one in the region. So as I mentioned, we collected our first sample in 2006. In 2012, we published a paper discussing the reason why it's important to have and collect, store information in a systematic way. We have tried our best and to maintain this registry since that time. So we started the Emirates Family Registry with a patient with diabetes. And it's now contained data from a range of different diseases. We have developed different sequencing analysis to allow us to improve the collection of information. We have also had to refine our research method. We are aligned with international standard to allow us to compete with international efforts. We would like to see this information that we have collected used in clinical setup in near future. So being unique and different defines our identity. However, it's possibility of little interest to others. Everyone is busy trying to define their own genetic identity to solve their local unique problems. Although I believe that we can learn from comparing different populations, the reality is that we do not tackle our issue locally and throughout the region. We will not have the intellectual capital to trade with others. So a local regional genome industry is important for our healthcare sector in the UAE and GCC. If we don't write our own books, basically nobody will do it for us. So thank you for listening, and I'm happy to take any questions.